Okay, last section of chapter 3. Let's go. All right. So, backing up to this one, interleukin 1 beta, which is going to lead to interleukin 6, and TNF alpha. These things are going to be, they're going to tend to come together. But with different diseases, you may have a different balance of these things. For example, in COVID-19, you'll have fairly low levels of interleukin 1 beta, really high levels of interleukin 6 wherever there's severe disease. And TNF alpha is basically going to follow interleukin 6. If you have a lot of IL-6, you'll have a lot of TNF alpha. And yeah, this is going to, it has a purpose, but when it's out of control, then it ends up being quite destructive. And so these things are going to be released, and they're going to have multiple functions in different parts of the body. Different functions in the liver, bone marrow, fat, dendritic cells, hypothalamus up in your brain. And they're going to cause different things. So they're going to mobilize neutrophils, or they're going to stimulate dendritic cells to go to the lymph nodes and mature. It's just a general all hands on deck danger signal that we are under severe attack, and yeah, basically everybody needs to get involved. And so fat is going to break down some energy to increase body temperature and basically, yeah, make it a little bit easier for the immune system and a little harder for whatever else is hurting you. And yeah, liver will also produce some other immune-related proteins, so that mannose-binding lectin that we covered in chapter 1, C-reactive protein that's going to be involved in inflammation, and yeah, they are going to activate complement, sticking to other things, which is called opsonization. And so we're going to fire up neutrophils. We're going to fire up the complement system. We're going to slow down, just try and throw off bacterial or viral replication. Bacteria and viruses usually operate best at a particular temperature. And that's usually going to be the temperature that they encounter most commonly. So it's going to be something like body temperature for things that are used to growing in or on us. And when you increase the temperature, you can actually cause a lot of these to sort of just break down. It's a kind of a neat thing. It's called temperature sensitivity. But a lot of bacteria, a lot of viruses have it. And so it's a general kind of thing that will help us and hurt bacteria at the same time, mostly. All right. So these cytokines, yeah, are for the most part going to be made by macrophages and to some extent dendritic cells. And so together they are going to make what we call the acute phase response, which is basically the first response. So this is the holding action. By that I mean like fighting in a way that doesn't necessarily end the infection but buys time for the immune system. And so macrophages are going to produce interleukin 6, for example, which is going to make liver cells produce all kinds of different factors, including some of the ones that cause clotting or that cause the normal and the alternative complement activation pathway. And then C-reactive protein is going to bind onto things on the bacterial surface and cause macrophages to be able to eat those a little bit better. So it all ends up working together. Because, again, the immune system is made up of all these cells, kind of working independently, but these messages give a little bit of cohesion, a little bit of a consistent message to the immune system, and generally, yeah, help us to fight things off. So these messages are the way that an immune system without any central controller is able to respond in a coordinated way to disease. You just got different cells with a wide range of different messages. And yeah, basically they end up getting the job done. It's like, I don't know if you've seen this, but there are some of these videos on YouTube where they'll have dogs talk by pressing little buttons. So they'll train a dog first with one or two buttons, like, you know, I'm hungry, or I want to go outside. And so then the person will ask the dog what it wants to do. So what would you like? And the dog will say, you know, hungry, yeah. And then the person will give them a little food. Yeah, and then, yeah. So the dog can learn this way. And yeah, there's some of these that have button boards that have 30 or 40 buttons on it that they seem to be able to sort of sensibly use as a kind of a language. these cytokines and chemokines work in the same way. They are the little button board that lets these cells, which are dumb as a bag of rocks, but it still lets them communicate just enough to be able to fight and destroy a variety of things that can go wrong in the body. Because remember, you're going to get viruses that you've seen before and viruses you haven't seen before in your lifetime. And for each one of these things, it's going to be brand new for your body, and it's got to find a way to fight against whatever the heck the world has thrown at you and win. So that's the neat thing about an immune system. It's kind of a little automatic defense system for your body. And so one of the ways that this works is by making interferons. So interferons are made very widely in response to viruses, also in response to some other things. But yeah, interferon alpha or interferon beta will activate a whole bunch of other proteins. So they're going to go through an interferon receptor. They're going to activate STAT1 and STAT2, which are going to eventually send a message into the nucleus and cause a change in expression. And that's something we'll talk about much later on in the class. We don't need to learn it now. Interferons will actually change the way that some of these proteins work. Things as fundamental as the ribosome can be modified because viruses come into your cells in order to use your ribosomes. And so, yeah, we can contribute to the host defense in a variety of different ways. So, there are several types of lymphoids, so actual white blood cells, that can have different functions early in infection. So these are going to be some of those white blood cells that do not actually change their DNA. So they're essentially part of the innate immune system, but they're going to be related to things like T cells. They're going to come from that lymphoid progenitor that makes T cells. So these are going to include things like NK cells, natural killer cells, and we're now starting to break down some of the other cell types. So these are things that would have been lumped in with natural killer cells or natural killer T cells at some point in the past. But the new way of breaking these down that lets us get at their function a little better is to call them innate lymphoid cells, or ILCs. And so there's type 1, 2, and 3 innate lymphoid cells, and they're going to be activated by different interleukins, so IL-12, 25, 23, 33, and we don't need to memorize these, but these are the functions of some of these things. They are going to come back, and we're going to see these over and over, and this is just an example for how some of these proteins work. They are all messages to some other part of the immune system, and so these are some of the messages and some of their responses. So some of these will cause NK cells to make interferon gamma, which is a nice message that'll go out to other white blood cells. It'll cause it to make perforin, which will actually perforate, punch holes in either your cells or other kinds of things. So this is a way to kill your cells that are malfunctioning or infected. And granzyme, which is another little destructive thing that can go in and root out other intracellular pathogens, for example. So how does this immune system, this innate immune system, start to work? Well, like I said, it's basically buying time for T cells to get fired up, make good T cell receptors to recognize which cells are infected, and then start to go out and kill. And it takes generally, I'd say about a week is a pretty good guideline for how long it takes to make an adaptive immune response. So in the first phase, you're going to have things like macrophages producing, and just general cells in the body producing interferons like interferon alpha and beta, or TNF alpha or interleukin 12. And then in the second phase, things like interleukin 12 are going to call in things like natural killer cells. And they're going to try their best to kill infected cells. But natural killer cells are not super smart. They're not super good at locating just the infected cells. They tend to kill other cells as well. And so you don't want these to be the sole basis of your immune response because it's just a bit too destructive, more than it needs to be. But they're buying time for you to make your T cell response. And yeah, this is a good rough approximation. So You'll have something like TNF alpha at around day 2. That's where it's going to peak. You're going to have natural killer cells peaking somewhere around day 5. And you're going to have your T cells coming online and getting to their early peak around day 7. And then with each kind of infection, it's going to be some version of this. Stuff's going to happen in the same order, but the different peaks can go on for longer or shorter times each. So yeah, natural killer cells are going to recognize healthy versus unhealthy cells. One of the things they look for is major histocompatibility complex class 1 protein. And remember, this is the protein that lets an ordinary cell show a little bit of whatever protein it's making at any one time. And so if a cell is not showing what it's making, then natural killer cells will rush in there and destroy that cell. Or potentially, if the target cell is making something that the natural killer cell can recognize, or if it can put out a special protein that's like a little flag that says, please kill me, basically, the natural killer cell will come in, bind to that, and just destroy that cell. It's got several different ways to destroy the cell, with its inhibitory receptor not being activated, with its activating receptor being activated, or with its trail receptor actually binding to the cell. And either way, you're going to end up with cell death if that natural killer cell gets activated, because that's what they do. They kill. They are natural killer cells. So natural killer cells, interestingly, have a big library of proteins that they can express. And so some of these things are like the KIR proteins. These are killer cell immunoglobulin-like receptors. And so these are all proteins that can be expressed on the outside of a natural killer cell and can allow it to recognize different kinds of danger. So think of it as a specialized surface-mounted version of something like the toll-like receptors or the nod-like receptors. It's just another way for natural killer cells to recognize exactly what it is that's making you sick and, respond to it. So we think of these as kind of maybe an intermediate version of the immune system. This is like showing maybe how an immune system could evolve from things that start out like you would find in more primitive organisms. They would just have things like the TLRs and the nod-like receptors, and that's their entire immune system. They'll just have more copies of them to recognize more things. Then you've got cells with like cell-mounted versions of proteins, and they're not related directly to those TLRs, but they're having the same function. So yeah, it kind of works like this. And so this will be your innate immune cells. We'll have stuff like this. And then the next step is to evolve a version of the innate immune cell, but that can customize its receptor. And that's going to be something like a B cell or a T cell. And we're going to call those the adaptive immune system. So there you go. That's how it works. And so these KIRs, for example, will be activating receptors. And if a KIR is able to bind to something on the surface of a cell, then boom, that is going to activate the natural killer cell. There are also inhibitory versions of KIRs to tell the natural killer cell when not to kill. So you can put out a don't kill me message if you're a regular cell, or you can put out a please kill me message. Or if you get infected by bacteria or a virus, maybe the cell loses its ability to put out the right message, and so that's fine. Natural killer cells recognize this, and they come in and they punish it hard. Yeah, so these are just some of the molecules that will actually sense those target cells that you would find on natural killer cells. And we don't need to memorize them, but there are a large number of these. And you can spend your life studying the natural killer cell part of the immune system. And you won't even end up studying all of these things, I think. You can spend a lifetime studying any couple of these things. Okay. The ligands for some of these natural killer cells proteins, like this one, NKG2D, are going to be things like, there's a whole family of proteins that cells can make to indicate that yes, I am sick, or yes, I'm healthy, or no, I'm not healthy, I'm sick, basically, and cause natural killer cells to come in and do the right thing, either knock out that cell or let that cell live. And so that's as deep as we're going to go into natural killer cells, because the big focus of this course is going to be on adaptive immunity, but we've got to get through our innate immunity. And so this is kind of where we draw the line. I think everything after from chapter 4 onward is going to be very much directed toward adaptive immunity instead of innate immunity. But yeah, these things they all have a role to play in keeping us safe. And if we only had adaptive immunity without this innate immunity, we essentially wouldn't be able to respond to anything for 7 days. And in those 7 days, you would probably be killed by whatever it is that you got, virus, bacterium, whatever. So, yeah, these are all useful parts of our immune system. So, I hope that made sense. At the end here, we have a list of all the videos. So, there's some wonderful videos that they put out on Vimeo, and I would encourage you to click on each one of these. Now, remember, if you get one of these links that's like this, it's going to put a thing called a non-breaking space between the 5 and the 9-4. So just copy this link and then go back and take out whatever is between the 5 and the 9-4, then paste it in, and then it's going to work. And it's the same for all of these. You can watch cells actually eating other cells, or you can watch the process there. Yeah, the inflammasome is a marvelous thing. This is a giant protein complex that's going to be involved in inflammation. Yeah, you got your little cytokine signaling, the leukocyte rolling. This is how these white blood cells, like neutrophils and monocytes, will actually roll along the inside of a vein. And when they come to the right signal, which is this rolling adhesion, then they will stop. And then they will actually migrate out of the bloodstream and into the cells. So that's the extravasation here. And so it's a mixture of nice little videos to help you understand the process and actual pictures of real cells doing the processes that we are describing here. So yeah, have a look at those videos. I think they'll help bring everything else into focus in Chapter 3. So there's a lot here. That's why we broke it down into 2 weeks. But yeah, I think it's worth it. I think there's a lot of good stuff to learn here. So thanks very much. This is finally the end of Chapter 3.